Welcome back, everyone. Next, we have Tharimmune Inc. It trades on the NASDAQ under the symbol THAR and is a clinical stage biotech company developing a portfolio of therapeutic candidates for inflammation and immunology. Please welcome President, Chairman, and CEO Randy Milby and the COO Sireesh Appajosyula. Nice to see you, gentlemen. Welcome to the conference today.
Thank you, Anna, and welcome, everyone. Thanks for joining the conference. I'm going to start out with a question. What do you do when you have an itch? Well, you scratch it. But what if it's an intractable itch? It's an itch that when you scratch it, it just never goes away and it continues. That's what we're going to talk about today. It's called pruritus. We will be making some forward-looking statements, but you can check out everything on our website, as you can see at the very bottom, tharimmune.com, and our 10K, which we just filed a week ago. A little bit about our background. I'll start with myself and then I'll turn it over to Sireesh. I'm a pharmacist by training. I was an ROTC person, so they paid my way through school, and then I served in the military for 4 years. After I finished my military obligation, I went back for an MBA at Washington University in St. Louis and started my business career at Bristol Myers Squibb in a management development program they had. I had a number of really cool jobs at DuPont Merck Pharmaceuticals and DuPont, both in sales, marketing, business development, licensing, etc. I served a stint at Goldman Sachs as an equity research analyst, and prior to this, I was the CEO of another publicly traded company called CorMedix. While I was there, we took the stock price from a market cap of $3 million to $350 million. I started this company in 2017, and Sireesh has actually helped me from the very beginning, but I finally convinced him to join full-time in July of last year. But Sireesh, please share your background.
Thank you very much, Randy. I'm Sireesh Appajosyula, the Chief Operating Officer of Tharimmune. I've been involved in biotech and biopharma for well over 20 years now. I've been a drug developer with operational experience, responsible for overseeing both preclinical and clinical stage projects. I've also had the experience to launch products into the United States market, and I've spent some time over the last few years at large organizations like Aventis, which eventually became Sanofi, as well as Salix Pharmaceuticals, which was acquired by Bausch Health, for about 10 years, where I have a deep experience in gastroenterology, hepatology, immunology, and oncology. Thanks, Randy.
Thanks, Sireesh. Everyone you see on this slide is a board member. So Leonard Mazur, he's the CEO of Citius Pharma. He has a lot of success at startups and exits. He helps me see around the corners, if you will. Lynn Boyce, physician, she's been with us from the very beginning as well. And then Kelly Anderson has a lot of successful exits as well. She serves as a chairperson of our audit committee. So what we're going to talk about today is, as mentioned, we're a clinical stage company, and TH104 is our lead. And it's for chronic pruritus, that itch, that intractable itch that I described earlier. And this is the itch affiliated with primary biliary cholangitis. I'll be calling it PBC in the future, but there's a high unmet need for this. We have intellectual property around this technology. We have the IND, and as you look on our website, we've completed the phase 1 study. I'll go a little bit into the CMC, the manufacturer, and in November, we raised $11 million. So indication for this, for 104, as you can see on the left-hand side, it's moderate to severe chronic pruritus in these PBC patients. The drug is nalmefene, and it's embedded in a proprietary transdermal buccal film. It's like a Listerine strip, but you just put it inside your mouth and it dissolves. And Sireesh will talk a little bit about the mechanism. It's the mu and kappa opioid receptors, and it's going to be given once daily. You can see the timeline as far as in the very middle. We have completed the phase 1 study. That PK bridging study, which Sireesh will share with you in a few minutes, and we're gearing up for the phase 2 study. The endpoint will be safety and tolerability, and the secondary endpoint, reduction of pruritus. So a little bit about this chronic pruritus, or this extreme itching in these PBC patients, as you can see. PBC is an orphan disease. Interestingly enough, there's the same number of patients with PBC in the United States as there is in Europe. But you can see what it is. PBC is actually an inflammation of the bile ducts, and a buildup of that bile causes extreme itching. Therefore, topicals don't work in this situation. What's interesting on the right-hand side, 65% of the patients, this itching is actually worse at night. many of them, and from interviews, they describe it as this intractable itch, like bugs crawling underneath their skin, where they just keep scratching and scratching. As you can see, since it's worse at night, the quality of life of these patients, 65% of these patients, is abysmal. Now we're focused on PBC. As you can see in this slide, there's over 4 million patients that suffer from liver disease in the United States alone. Over half of these patients have pruritus. Now we're focused on PBC because 70% of these PBC patients are affected by pruritus at some point in their disease, as you can see. But on the right-hand side, you can see pruritus in an assortment of liver diseases. But again, we're focused just on PBC at this point. And that serves as the foundation, as you can see on slide 8. So you can see that 40% of these chronic pruritus and liver diseases, that's that 1.7 million patients that I talked about in the previous slide. So quick math here. If 50% of these patients, half of these patients were treated with our drug, 500,000 some patients treated with the drug. Talking with the key opinion leaders, they say each patient would probably, they felt we should price it between $8,000 to $10,000 per patient for a year. So you can see the market potential for this just in liver disease alone is $350 to $500 million opportunity. Now that's just in liver disease, but as you can see on this build, pruritus is also a bad actor in chronic kidney disease and obviously at the very top, atopic dermatitis. But here again, we're focused just on the liver disease. This is something I always tell the team, we have to get one through the keyhole first. And PBC, chronic pruritus and PBC is our first. I talked a little bit about the CMC on one of the earlier slides. As you can see, this is the size of a dime. So what happens is the intellectual property is this micronization of nalmefene on this proprietary transmucosal film. The patient attaches this to the inside of their cheek in the evening, and it's absorbed within 10 minutes. But the interesting point about this, why is this different than others? You can see at the very lower right-hand corner is because this, by using the transmucosal delivery, this buccal delivery, you're actually bypassing the liver. Therefore, you don't have a first pass effect. So the drug is actually absorbed faster and distributed to the skin faster. So that, as I mentioned, it's absorbed within 5 minutes, and it peaks in the blood level, it peaks in the blood 30 minutes after attaching it to your cheek. And this is very important because we'll give this in the evening, 30 minutes before bedtime. As I mentioned on that earlier slide, nocturnal pruritus for 65% of those patients. So the patient applies this daily at night, 30 minutes before bedtime. On the left-hand side, you can see how it's manufactured. As I mentioned, the intellectual property is around the film itself and also around the micronization of the nalmefene on this film. At this point, I'm going to turn it over to Sireesh, and he's going to share some of our data with you. Sireesh?
Thank you, Randy. So our product, known as TH104, which Randy gave you the background on, has a robust preclinical package, meaning information that is required in order to submit to the FDA what's called an investigational new drug application, which the company received last year. This application allows for human studies in the United States. The studies you see on the slide here are important because you can see on the left in animal models, as Randy just detailed, you see a plasma concentration increase of the active drug. We can detect it in 5 minutes. You see that at 15 minutes, it starts to get much higher. And as Randy mentioned, peaks at 30 minutes. And the absorption being detectable in 5 minutes allows us to think about the fact that this can act pretty quickly as it gets absorbed through the buccal cavity or inside of the cheek. The graph on the right compares our buccal formulation to an intravenous injection of the active drug. So this is an approved drug that's being tested against our formulation, which is a buccal formulation. The intravenous formulation is an acute indication, meaning that it can only be used one time. But what's important to see here is that we achieve high bioavailability in an hour of dosing. And when the drug concentration declines, it's matching the IV dose over 12 hours. This allows us to be confident that TH104 can be dosed once a day while achieving a fast onset of action and has predictable blood concentrations. Because we have an IND, we were able to do human studies, and I want to share a few with you right now. The graphic on the left looked at our drug TH104 in healthy volunteers, and the aim was to predict the pharmacokinetics or blood concentration of the drug when compared to an oral tablet formulation that is only approved in Europe, not in the United States. But it's important for us to test against multiple formulations to see what benefit we can gain from a buccal delivery. And as you can see on the left side of this slide, the concentration mimics the oral drug. In fact, we have slightly higher exposures, meaning a calculation called an area under the curve shows that we have higher exposures of our active drug compared to the oral tablet. This is an advantage because the oral tablet has to go through liver metabolism as its concentration rises, whereby TH104 bypasses this effect. And this is important because we want to have high concentrations of the active drug in the blood so it can do the work it needs to do in order to reduce the pruritus or itchiness in these patients. The graph on the right is simply two different formulations of different pH or acid formulations that are applied to the cheek. And the higher concentrations are achieved with a neutral formulation, which is good news for the clinical program since we don't want to irritate the buccal cavity and we want to mimic the neutral pH of normal mucosa in the mouth. Also important, at the bottom, we did not see any new adverse events here in the healthy volunteers that were dosed. Most of the side effect profiles were mild, and the highest event was somnolence or sleepiness, which we already detailed in an earlier slide by Randy, which shows that this program could be dosed in the evening at bedtime because so many of the patients complain about worse itching at night, known as nocturnal pruritus. Now let's look at another study that looked at actual liver disease patients. This was a trial that was conducted as a single-dose, single-center, open-label randomized trial of a low dose of our product, TH104. And this looked at two different cohorts of liver patients, mild liver disease patients, which are categorized by hepatologists as a Child-Pugh A liver scoring system cohort. It simply means they have liver damage, but it is not moderate or severe. Child-Pugh B is another liver cohort calculation whereby they look at the liver and it's a much more severe disease. So both of those types of patient profiles were enrolled in this phase 1 chronic liver disease trial. The blue bar and the orange bar denote those two different types of liver damage, and the gray bar denotes all of the patients in totality. This looked at the buccal strip at a low dose, as I mentioned, as one dose. And the endpoint that we wanted to look at was safety and tolerability, of course, but also we wanted to test what this debilitating itch looked like in these patients using a validated endpoint that has been used for other drugs. And this is known as the worst itch numerical rating scale, or WI-NRS. It simply means it's a scale from 0 to 10 that the patient is continuously assessing throughout the entire day. And as you can see, from 1 hour all the way to 24 hours, there was a consistent decrease across the two cohorts, as well as the total enrolled in this trial, whereby we showed a 33% decline in itch scores after a single dose at 24 hours. This was very good proof of concept for us that our drug is getting to where it needs to go to be able to lower the itch scores. We will be looking at this and many other parameters in a phase 2 trial that we hope to launch later this year. I want to talk a little bit about the mechanism of what our formulation and this drug is doing. It's been well known that changes in the expression of opioid receptors in the body and their interaction with their natural ligands is a balancing act, meaning that in certain diseases, including liver and atopic diseases, there's an imbalance that could occur. And the cartoon on the left side of this slide denotes that. It shows that where one type of opioid receptor may become overmodulated. And so data suggests that there is this itch circuitry in the body, and it has a lot to do with the imbalance between two types of opioid receptors, known as kappa and mu receptors. The drug we are using in this unique formulation is a unique drug because nalmefene corrects this imbalance by restoring the body's itch circuitry back to a normalization. And it rebalances this dysregulation because the one drug affects both receptors. And that's why it makes it ideal to help patients with chronic pruritus, specifically in liver disease. The middle part of the slide is data which shows normal endogenous opioids that are secreted by the liver in chronic liver disease patients. Because the liver is impaired, it's trying to compensate by releasing more of these natural opioids to decrease pain and also improve patient well-being for the patient. But unfortunately, the unintended effect of these secretion of endogenous opioids is that it activates this itch sensation I just discussed. Furthermore, we know that nalmefene is an ideal compound because of previously published data that's on the far right of this slide, which looked at oral tablet doses, which are not available in the United States, but they looked at very high doses as they have to go through the liver, as high as 240 milligrams twice a day. But we know the drug works because in that study, they showed a 75% reduction in scratching activity, as well as in itch perception in patients with primary biliary cholangitis. It's yet another piece for proof of concept that we have the right strategy with the drug, but we need the formulation to be improved, which is the buccal formulation. I want to touch quickly on one more mechanistic study. The graph on the left, and in fact the one on the middle, show an increased amount of interleukin-17. On the left, it shows interleukin-17 increases in liver tissue of primary biliary cholangitis patients. And the graph in the middle shows increased interleukin-17 in the serum or plasma in PBC patients. This is important because interleukin-17, or IL-17 as it's often referred to, is an inflammatory cytokine. It has been well studied for many years, and it has been linked to many different inflammatory conditions. The data on the right-hand side of the slide shows that nalmefene, in animal models at least, has anti-IL-17 effects. And we find this quite interesting, and we'll be looking at this in clinical studies as we progress the program. Because we know that opioid receptors are involved in itch perception, but IL-17 may be involved in inflammation. And if we can decrease both of those, we believe that we will have real mechanistic evidence against PBC itself as well as pruritus. Now let me focus on what we just completed recently and what the FDA asked us to do. The FDA asked us to conduct a phase 1 study in the United States, which was recently completed. The study design is a crossover design where patients are dosed with a single dose of our buccal strip and compared to an intravenous nalmefene single dose that's approved in the United States. After a 7-day washout period where we look at serial blood concentrations over time, they're crossed over to the other group for the drug they didn't receive the first time, and we repeat the serial blood concentrations. We've already reported that this was a safe and tolerable study with a good safety profile compared to the IV drug for our product. We will continue to analyze this study and present the pharmacokinetic evidence compared to IV nalmefene with TH104 in the early second quarter. After conducting this study and providing evidence to the FDA, we will begin to start planning and are already planning for our phase 2 program. I'm going to turn it over to Randy now to talk about the anticipated milestones over the coming months. Thank you.
Thank you, Sireesh. As you can see on this slide, these are the activities that these are the inflection points as I describe it. As Sireesh just mentioned, we've completed the phase 1 bridging PK, and we'll have the final bioavailability data out this quarter. And you can see that we're going to start in the third quarter our phase 2 initiation of the PBC. So this will be patients with PBC using the buccal film. And we'll have the top line results from that either late fourth quarter of this year or first quarter 2025. What you can see in the light gray in between, these are activities that are going to be completed as we advance this technology. These are the key inflection points or the milestones over this next year. So you can see in 2024 is a big year for us as far as completing a lot of the completing the phase 1 and phase 2, and then gearing up, as you can see, in the second quarter of 2025, discussing with the FDA that end of phase 2 meeting and preparing for the registration study. This slide is just a summary. So you can see on the lower right, we just went through the timeline. And we talked earlier about this chronic pruritus, this debilitating, it's uncontrolled itching, like the patients described, like bugs crawling underneath their skin. We're focused on PBC patients because 70% of those patients are affected by this chronic pruritus. And as Sireesh mentioned, 65% of the patients have this nocturnal pruritus. The technology is like a Listerine strip. As you can see, it's the size of a dime. The intellectual property relates to both the film itself, as well as the micronization of the nalmefene, which has been proven, and it's attached to the inside of the cheek once daily. We feel it's de-risked because Sireesh shared with you a lot of the data that's already available on nalmefene itself and the data that's been done outside of the United States. We have that IND, we completed the phase 1 study, and we're getting ready, as Sireesh just mentioned, to start the phase 2. This chronic pruritus and PBC, as I shared earlier, it's PBC initially, but we can expand into other liver diseases and inflammation, pruritus diseases as well. I showed you the build slide of liver disease only, which is over 4.4 million patients in the United States have liver disease, 1.7 suffer from pruritus, but we also talked about the chronic kidney disease as well as atopic dermatitis. The drug which we shared with you is nalmefene, and it's delivered on this transmucosal film product, which is intellectual property around the film product as around the nalmefene itself, the way it's micronized. And you can see the CMC, it's de-risked because this is technology that we've already had in man, and we have intellectual property. Our philosophy on IP is we'll continue to build a picket fence around this intellectual property as we continue to advance the technology. In summary, we raised, as I mentioned at the very beginning, $11 million in November. If you look at our 10K, at the end of December, we had $10.9 million in cash. Going over to looking at the right-hand side, that amount of cash allows us to complete the phase 1 study and complete the phase 2 study and takes us into the first quarter of 2025. Total common shares, stock outstanding, 11.7 million shares, and number of warrants is minimal, just over 300,000 warrants with an exercise price of $4.50. That concludes our formal presentation. I'll turn it back over to Anna now for questions and answers.
Great job, gentlemen. All right, we do have some questions for you. Let's see. Sorry, I got off on the. All right, let's start with. Here it is. Sorry. Okay, topicals. Any actually prescribed for PBC? Can you talk a little bit more about the topicals?
Sure. Sireesh, why don't you share what's currently being used in PBC?
Yeah, there are a lot of things that are tried. Topicals are certainly tried. Corticosteroid creams, which have their own issues as far as getting absorbed into the skin. but also antihistamine type of topical products, lotions, calamine lotion, which we're all familiar with, that folks use for poison ivy, et cetera. Like a lot of the topical things are tried, but unfortunately, none of those things provide relief, because this is not a condition where the pruritus is arising from outside of the body, where it's an irritant that's causing the itching. This is happening because the liver, which has bile ducts inside of it, are dysfunctional, and the bile is actually spilling into the bloodstream. And that is an internal systemic issue that needs to be tackled with drugs that could target the internal system and not externally on the skin.
And Scott has a follow-up question with that. Just to be clear, your technology is meant to treat the itch and not the ailment causing it, like you just mentioned?
That is correct.
Okay. Eric Ribner asks, how are you different than Trevi?
Sireesh?
So Trevi uses a different kind of a compound, and they use nalbuphine, which is a different sort of a opioid agonist antagonist qualities. So we've assessed all of the different compounds that affect both of those receptors that I talked about, the mu and the kappa receptor. And the qualities of nalmefene itself are quite important. It has a very long half-life. It affects the mu and kappa receptors sort of in the correct way in order to restore that balance that I talked about earlier. So all of these opioid antagonists have slightly nuanced differences for what you're trying to treat them for. And for treating liver-related itch in this specific formulation, as you try to bypass the liver, nalmefene is the ideal product, because there is lots of data available that has previously been done, not with this formulation, but with the drug itself, that lends us real good proof of concept that nalmefene is the ideal drug for chronic pruritus.
And Tracy Goodwin asks, how often is it applied, and what will the cost be for each dose?
It's applied, as we've talked about, 30 minutes before bedtime. So it is for chronic therapy. So every day the patient will apply it, attach it to their cheek each evening, 30 minutes before bedtime. And as far as the pricing, we haven't come to the final numbers yet, but when we talk to key opinion leaders, particularly in this space, as I mentioned earlier, they're saying that $8,000 to $10,000 per patient per year.
And Chris Bates wants to follow up with that $10,000 per patient. Is there a point that you could price yourself out for what is covered by insurance companies, or are there always negotiated rates? And what do you think will actually be realized for an application?
Well, Sireesh has done a lot of work in this space. I'll turn it over to Sireesh, his thoughts.
Yeah, I think it's just a bit too early to get real nuanced about pricing. I think the numbers that Randy told you are quite reasonable for this disease, but really what this depends on is things beyond sort of the phase 2 and phase 3 development as far as what clinically meaningful relief means for these patients. You know, how much itch decrease can we get to? We've shown on one dose that we can get to 33.3%, which is 1/3. On a chronic dosing, we'll see what those numbers are later in the phase 2 trial. But really, this is really about proving to pharmacy benefit managers, et cetera, the burden of illness of this disease and all of the things that go into what is causing them to have missed days at work, missed days at school, missed college days, et cetera. So all of those calculations go into how effective these people can be with a drug that can really relieve them of this intractable, unrelenting itch for which there is no treatment. And so proving all of those things is going to take some time, which is what we do in this drug development process. But I think it's a bit too early to talk about sort of what would be covered and what wouldn't. But there's lots of data to suggest that this type of a product is really a high unmet need in this marketplace. And those negotiations will start to happen as we get a little bit more data into phase 2, but we feel pretty comfortable about that.
And Kurt Boyd wants to know, how will the drug be classified?
Classified meaning if you're talking about a controlled substance, because we talked about opioid antagonism, et cetera, these are antagonizing the opioid receptor. So they are the opposite of opioids, meaning that they displace opioids off of the receptor. So there is no, these are not controlled substances.
And Jake Mathis wants you to talk a little bit more about the phase 1 and 2. What is to be anticipated once it's completed, and when will it be completed?
Go ahead, Sireesh. You can highlight again your phase 1 data, particularly the data output, and also the timing and what we're looking for in that phase 2 study.
Yeah, so the phase 1 was completed. It looked at 20 patients, and it was a crossover design, as I mentioned. And that looked at both the buccal film, which is TH104, and it compared to the intravenous nalmefene injection, which is used in the United States for opioid overdose. So it's a single-use injection. So the FDA asked us to compare it to what's known as a reference label drug. And the reference label drug for nalmefene in the US is the IV injection. So we completed that, and we released, we put out a press release recently that talks about the safety and tolerability. Our drug was safe, tolerable. We saw no sort of new adverse events that we hadn't already seen. They were all mild, and they were very comparable to what we had always seen with the drug. So that's good news. What we're waiting on now is the bioanalytics, which is the levels of nalmefene in the blood and some of the metabolites of nalmefene, which we have stated would be part of our top-line readout, which we will read out in the second quarter. We believe that could be in the early second quarter, which is not too far off. As far as the phase 2 trial goes, the phase 2 trial is going to be looking at actual PBC patients. We have some good proof of concept based on data I showed you in an earlier slide that looked at the one single dose, as I mentioned. But these primary biliary cholangitis patients, we have not publicly stated sort of what that trial design will look like. So stay tuned for that, which we will be doing shortly. We're having lots of discussions with FDA around what that looks like. But I can tell you that it's probably going to be somewhere in the less than 40 patient range total. It will be placebo controlled. It will be multiple doses. We've stated that it will probably be a 4-week study. So we're going to dose patients for much longer than we have. And then we'll really get an idea around the itch decrease over time. And the important piece of the phase 2 trial, which I think the question was, when will it be completed? And Randy touched on it, potentially based on enrollment late fourth quarter, but certainly in the first quarter of next year, we'll have that phase 2 top line data. But that data is going to be the proof of concept that will compare between placebo and our drug, what the difference in itch perception could be, the decrease in itch. And that's going to give us really good data to be able to calculate what the placebo difference could be that allows us to plan for a potential registration trial, which can then be filed for approval with the FDA.
Wonderful. Well, thank you, gentlemen, for your time and presentation today. Really important work you're doing. We appreciate you joining us on the conference.
Thanks everyone for joining.
Thank you for having us.
All right. Stay with us, everyone. We'll be right back with a Septoscope.